DNA Depot presents How Safe is Our Food? Kit DD206. In this experiment, you will discover how bacterial food contamination is a growing problem worldwide and how it is detected by the use of colorimetric indicators. You will also learn about common bacteria that can contaminate food and cause illness. Your kit should include chloroform detection broth, chloroform control cells, nutrient broth, test tubes with caps, sterile calibrated transfer pipettes, plastic conical tubes with caps, and wooden sticks. You'll also need samples from food sources such as salad greens like lettuce, spinach, or green onions, disinfectant, a test tube holder, a plastic container to soak used sample tubes, a laboratory marking pen, disposable laboratory gloves, goggles, and an incubator. Experimental procedure: preparation of chloroform control cell suspension. This must be done 1 hour to 1 day prior to starting the experiments. 1. Use a sterile transfer pipette and add 2 milliliter sterile nutrient broth to the vial of chloroform control cells. 2. Replace the rubber stopper and cap. Mix gently by inverting until the freeze-dried plug is dissolved. 3. Incubate the vial of cells for 30 to 60 minutes in a 37 degrees Celsius incubator or overnight at room temperature. Preparation of samples and negative control. 1. Place 6 small test tubes in a test tube holder. 2. Carefully open a bottle of chloroform detection broth. 3. With a sterile pipette, aseptically add 1 milliliter of broth to 5 tubes and 2 milliliter of broth to 1 tube. Cap the tubes. 4. Label the tube with 2 milliliter broth negative control for the negative control sample. Incubate overnight in a 37 degrees Celsius incubator or for 48 hours at room temperature. 5. Save the other tubes for testing food extracts and positive control. Preparation of positive control sample. 1. Take one of the tubes with 1 milliliter chloroform detection broth and label it positive control. 2. Use a sterile pipette to transfer 1 milliliter of the chloroform control cell suspension to the tube labeled positive control. Soak the pipette in 10% bleach for 20 minutes before discarding in regular trash. 3. Cap the tube and incubate overnight in a 37 degrees Celsius incubator or for 48 hours at room temperature. Preparation of the vegetable extracts. 1. Cut up vegetables such as lettuce, spinach, or green onions into small pieces. 2. Place pieces of cut up vegetables into a large conical test tube. 3. With a transfer pipette, add about 1.5 milliliters water to the vegetable. 4. Use 2 wooden sticks to mash the vegetable in the tube until wilted. 5. Label the tube with the name of the vegetable. 6. Repeat steps 1 through 5 for 3 other vegetable samples. Test for bacteria in vegetable extracts. 1. Label the 4 tubes with 1 milliliter chloroform detection broth with the names of the 4 vegetables extracted. 2. With a transfer pipette, add about 1 milliliter of vegetable extracts to the corresponding tubes. Use a new pipette for each vegetable extract. 3. Cap the tubes and place in the test tube holder with the positive and negative control samples. 4. Incubate tubes in an incubator at 35 to 37 degrees Celsius for 24 hours, or at room temperature for 48 hours. Food contamination can be introduced at various stages of food processing, so the next time you buy salad greens from the supermarket, make sure you wash them carefully, even though the package might be labeled pre-washed. Cook meats well and make sure that you wash your hands and the cooking utensils, as well as the kitchen countertop.